Today's topic is immunochemistry. We are exposed to a wide variety of pathogenic microorganisms which can cause serious diseases. Nature has equipped us with highly competent defense mechanism to deal with the foreign invaders. The defense mechanism comprises innate immunity, acquired immunity. So the question arises that what is innate immunity? Skin and mucous membrane act as a physical barrier. Secretions covering skin and mucous membranes create an inhospitable environment for pathogenic microorganisms. Phagocytes, which includes macrophages, neutrophils, etc., engulf and destroy the pathogenic microorganisms. Complement proteins, acute phase proteins, and interferons, etc., help in destroying the pathogens. In contrast to adaptive immunity or innate immunity, does not recognize antigens. It is not. It is non-specific and does not improve on repeated exposure to the pathogen, same pathogen. What is adaptive or acquired immunity? Adaptive or acquired immunity comprises of humoral immunity, which operates through circulating antibodies, which recognizes and act against extracellular antigens. Cell-mediated immunity, which operates through activated lymphocytes, which destroy the antigens that have entered some cell. Now, what are antigens? Basically, all the foreign chemicals having high molecular weights, protein, polysaccharides, synthetic chemicals, complex structures. They may enter as free molecule or as a component of some cell. They can evoke an immense immune response, humoral or cellular or both. A small part of antigen known as antigenic determinant or epitope is recognized by the immune system. What are haptens? They are low molecular weight proteins, cannot evoke an immune response by themselves. They can evoke an immune response which combine with some other large molecule. Once the immune response develops, the free hapten can be recognized by the immune system. What are the role of lymphocytes? Lymphocytes, they play a key role in immune system. They are one of the most important cells of immune system. They are formed from stem cells in bone marrow, differentiated into B lymphocytes, B cells, or T lymphocytes, or T cells. B lymphocytes are processed in bone marrow, that's why the name B. And T lymphocytes are processed in thymus, so T lymphocytes. T lymphocytes synthesize antibodies that provide humoral immunity, and T lymphocytes provide cell mediated immunity. Both are capable of recognizing antigens. A given cell can recognize only a specific antigen. What is humoral immunity? Humoral immunity operates through antibodies or immunoglobulins, which are proteins for defense mechanisms. The basic unit of an immunoglobulin or Ig consists of two heavy chains of amino acid joined to each other and one light chain joined to each heavy chain by disulfide linkages or disulfide bonds. This is the structure of an Ig or an immunoglobulin. We can see here in the blue. These are the heavy chains, and yellow, these are the light chains. There are two terminals as are in proteins. The N terminal or the amino terminal is at this fork-like structure, and C terminal is at the base of the antibody. Immunoglobulins or Igs, their light chains have a molecular weight of around 23,000. There are two types, kappa and lambda. Heavy chains have molecular weight of around 50,000 to 70,000. And they are of five types, alpha, delta, epsilon, gamma, and mu. A given immunoglobulin has only one type of light chain and one type of heavy chain. In one type of light chain, say kappa or lambda, the C terminal half has a constant amino acid sequence and N terminal half has a variable amino acid sequence. In one type of heavy chain, the C terminal 3/4 has a constant amino acid sequence and N terminal has 1/4 has variable amino acid sequence. The chains are folded to form some globular domains. The variable region of the light and heavy chains comes together to form antigen binding sites. These are the sites which are capable of forming a bondage with the antigen and thus known as antigen binding sites. We can see the variable and constant region in the heavy chain and in the light chain both. This is a molecular structure of an immunoglobulin. We can see here the variable region of light chains are and heavy chains have three hypervariable or HV regions in which the amino acid sequence is extremely variable. The hypervariable region constitute the antigen binding sites. The hypervariable regions are also called the complementary determining region or CDRs, as they determine the complementarity between the antigen and the antibody. We can see here the hypervariable or complementarity determining regions or CDRs, along with the disulfide linkage between the heavy and the light chain and the hinge region. Immunoglobulins, only a small region of the antigen known as epitope is recognized by the antibody. The region of antibody that contacts with epitope is known as a paratope. So antigen has epitope, which is an end of an antigen, which is capable of binding to the antibody, and the part of antibody which binds to the antigen or epitope is known as a paratope. Now we see the action of papain. The proteolytic enzyme papain cleaves the antibody into two identical Fab fragments and a FC fragments. We can say FAB or FC also. Each Fab fragment can bind antigen, fragment antigen binding. An FC fragment can be easily crystallized, known as fragment crystallizable. Papain treatment has thus shown functions of different regions of antibody. We can see here antigen binding sites and then again see the papain cleavage, which causes the hinge joint or the disulfide linkage of the antibody. And so the antibody is cleaved into two parts, the Fab fragments or the upper part and the FC fragment or the lower basal part. The antigen and antibody interactions are very specific in nature. As I said earlier, that antigen, there are particular antigen, a particular antibody will be formed against it. The antibody binds the complementary epitope of an antigen by hydrogen bonds, electrostatic bonds, hydrophobic interactions, and van der Waals forces. The binding is non-covalent and reversible, so it is not very strong since it is not non-covalent. It is not covalent. After antigen binding, the constant region of the antibody performs the effector functions. What are the effector functions and mechanism of it? The antibodies defend the organism by a number of effector mechanisms known as neutralization, opsonization, and complement activation. What is neutralization? Antibody coats the antigen on the surface of the pathogen and prevent their entry into host cells. Antibodies coat the toxin released by the pathogen and prevent their toxic effects. Antibodies coats the surface of the pathogen and facilitates phagocytosis by attracting phagocytes. This is known as opsonization. Antibodies also attract natural killer cell or NK cell. The NK cell causes antibody-dependent cell-mediated cytotoxicity known as ADCC, which kills the pathogen. The last is complement activation. Binding of antibodies to the antigen on the surface of pathogen can activate the complement system. Complement system either destroys the pathogen directly or facilitates their destruction by phagocytosis, which is a process of engulfing the whole antigen by a lymphocyte or a monocyte. The classification of immunoglobulins. Immunoglobulins are classified on the basis of their heavy chains into 5 classes. Immunoglobulins A, immunoglobulin A, IgG, IgD, E, and M. Their heavy chains being alpha, gamma, delta, eta, and mu. The light chain being kappa or delta. There are subclasses further. IgA has 2 subclasses, IgA1 and IgA2, and IgG has 4 subclasses from 1 to 4, IgG1 to IgG4. We see the first immunoglobulin, which is immunoglobulin G, the most abundant in plasma, longest half-life among all the immunoglobulins, performs all the effector functions, can cross the placental barrier, and maternal IgG provides immunity to the newborn in the first few weeks of his life. You can see the structure of IgG. IgA is present in plasma and is in exocrine secretions. IgA presents its exocrine secretions. IgA present in exocrine secretions is known as secretory IgA. Secretory IgA is formed beneath the basolateral surface of epithelial cell. It binds to an IgG receptor present on epithelial cells. A part of the Ig receptor detaches from cell and attaches to IgA dimer as secretory component. Secretory IgA transfers the cell to reach mucosa, where it acts mainly on the process of neutralization. Secretion of IgA is present in breast milk, which enters the gut of the newborn and protects against gastrointestinal pathogens. We can see the structure of IgA monomer and the secretory component of IgA, which has a J chain. You can see here basically there are 2 IgA molecules combined by a J chain. Third one is IgM. It is the largest in size, the first Ig to be secreted upon entry of any antigen, acts mainly by complement activation, weak neutralizing and opsonizing activity it has. This is the structure and it is the pentameric IgM structure. Where we can say the J chains combined together. IgE is bound to mast cell and basophils. Upon binding parasitic antigen, mast cell release the chemical mediators that destroy the parasite, also mediates in allergic reactions. You can see the structure of IgE. IgD is the last one where the functions of IgG is not very clearly known. These are the important features of different classes of immunoglobulins. We can see the plasma concentration here of different classes. Among them, the IgG has, since I said earlier that it is the most abundant one, so 700 to 1,500 milligram per deciliter, its distribution. Among all these, the lowest is of IgE. Half-life in days is IgG has 23 days, 5 to 6 and 5 to 6 of IgA and M. IgE and D have a half-life of 2 to 3 days, and they are mostly active against certain set of pathogens. So bacteria and viruses, for bacteria and viruses, IgG is there. For antiviral activities, IgA is there. For parasite, IgE is there. And since I told earlier that IgD, its function and its antigens are not known properly. So there are some variants of basic immunoglobulin structures. These are being idiotypes, isotypes, and allotypes. We'll discuss these further. Idiotypes, the variable region of an immunoglobulin forms a typical three-dimensional structure complementary to the antigenic determinant of a particular region. This typical antigen recognition structure constitutes an idiotype. We can see the structures of idiotype here that they are colored. The isotypes are immunoglobulins are divided into five classes: IgA, D, E, G, and M. Now, IgA is further subdivided into IgA1 and A2, which are basically the isotypes of IgA. And earlier, as I told you, that IgG has four subclasses: IG1, IG2, IG3, and IG4. These variants of classes and subclasses are known as isotypes. Isotypes, isotypic variants are due to differences in amino acid sequences in the constant region of heavy chains. You can see here the constant region of heavy chains have isotypes. Last one is allotypes, the mutation leading to substitution of one or two amino acids in the constant region give rise to immunoglobulins of the same class or same subclass, differing slightly in amino acid sequences in the constant region, known as allotypes. Allotypes are inheritable and they are inherited from parents. These are basically type of antibodies which are passed from parents to the offsprings. You can see here the green they are shown here. The next topic is antibody diversity. Our immune system can produce a vast range of antibodies. These antibodies can recognize virtually every actual and potential antigen. Antigen binding site of every antibody is unique due to a unique amino acid sequence in the variable region. According to one gene, one protein theory, every protein is encoded by one gene. Theoretically, millions of genes are required to encode the huge repertoire of antibodies. However, the number of immunoglobulins gene in human genome is less than 200. So only there are 200 genes which code for a protein which becomes an immunoglobulin or an IG. How a small number of genes produces a wide range of antibodies remained a mystery until Tonegawa shows that antibody diversity arises from gene rearrangement. This is known as light chain gene rearrangement. Genes from light chain and heavy chains are present on different chromosomes. A light chain gene is made up of three segments: variable, joining, and constant, known as V, J, and C segments, respectively. Genes for these segments are present in three different clusters of chromosomes. The gene for kappa light chains are present on chromosome 2. There are 40 genes for V segment, 5 for J segment, and a single gene for C segment. The genes for delta light chains are present on chromosome number 22. There are 30 genes for B segment, 4 for J segment, and 4 for C segment. The B segment and J segment genes encode the variable region of the light chains. The C segment genes encode the constant region of light chains. When a B cell differentiates on one of the B segment genes, join one of the J segment gene, and the intervening DNA is deleted, which is known as VJ joining. The VJ combination and a C segment gene are transcribed to form an hnRNA. hnRNA is spliced to form mRNA for a light chain. You can see here the V joining and gene rearrangement, further leading to the production of transcription of basically an mRNA, which is a light chain mRNA. And this further comes out from the nucleus and binds to the endoplasmic reticulums and then further to the ribosome, leading to the production of proteins or polypeptide chains, which forms an antibody. Since there are 40 genes for B segment and 5 genes for J segment, only kappa light chain 200 different BJ combinations are possible. Since there are 30 genes for B segment and 4 genes for J segment of delta light chain, 120 different combinations are possible. A given B cell has one particular BJ combination. Heavy chain gene rearrangement. After light chain gene rearrangement, we see the heavy chain gene rearrangement. A heavy chain gene is made up of 4 segments. The variable diversity joining in constant segment, known as VDJC segments. These genes are present in 4 different clusters of chromosome 40. There are 40 genes in the V cluster, 25 in P cluster, and 6 in cluster J, and 9 in C cluster. When a B cell differentiates, one D segmented gene joins one J segment gene and intervening DNA is deleted. This is known as DJ joining. One B segment gene joins DJ with deletion of intervening DNA. And VDJ combination and the first C segment gene are transcribed to form hnRNA. The hnRNA is spliced to form a heavy chain mRNA. VDJ join portion encodes the variable region, and there are 6,000 different VDJ combinations possible. A given B cell has one particular combination of VDJ combination. Similar to the light chain gene rearrangement, we can see here the first gene rearrangement, which is known as DJ joining, and the second gene rearrangement, which is known as VDJ joining, and further transcription and splicing to form mRNA, which codes for a heavy chain of the immunoglobulin. Thus, several hundred light chains can be formed, each having a unique amino acid sequence in the variable region, and several thousand heavy chains can be formed, each having a unique amino acid sequence in the variable region. When light chain combine with heavy chain to form an immunoglobulin molecule, millions of combinations are possible, each specific for one antigen. A given B cell has only one combination and forms only one particular antibody. What is combinatorial diversity? Antibody diversity generated by the variable recombination of DNA of different gene segments is known as combinatorial diversity. Nearly 2 million valuable regions can be formed by combination diversity. Gene rearrangement reactions are catalyzed by the usual DNA repair enzymes and two protein encoded by recombination activation genes 1 and 2, known as RAG1 and RAG2. The junctional diversity, a second source of diversity is junctional diversity. At the time of joining, some nucleotides may be added or deleted from the ends, creating a new sequence. Nucleotides are added by terminal deoxyribonucleotide transferase or TDT, and nucleotides are deleted by exonucleases. The third one is somatic hypermutation. A final source of diversity is somatic hypermutation, where a mature B cell encounters an antigen, a series of point mutation occurs in the variable region of the gene, resulting in further diversity. Like other genes, the immunoglobulin genes are preceded by a promoter. A regulator is present upstream on the promoter. We can say here the structure of a heavy chain gene, which has a regulator, a promoter, a variable region known as VDJ joining, enhancer, switch sequence, and constant region. An enhancer element is present downstream of the variable region known as VDJ segment or region. The first constant region gene is C mu. Each constant region gene is preceded by a highly conserved short repetitive sequence known as switch sequence. The promoter is weak, but gene expression is increased by enhancer elements. There are 9 constant segment genes for heavy chains. After VDJ joining, VDJ is transcribed with the first constant segment of gene, which is C mu. IgM is the first immunoglobulin to be formed upon entry of any antigen. Different classes of immunoglobulins differ in their heavy chains and their effector functions. IgM is the first immunoglobulin to be formed. However, Ig of a different class may be needed to deal with same antigen. For this, the BDJ complex already formed joins a downstream constant segment gene with deletion of intervening DNA. Thus, an Ig of a different class but having the same antigen specificity is formed. This is known as class switching or isotope switching. Switch sequences facilitate class switching. Lymphokines released by T cell also have a role in class switching. Differentiation of B cell into plasma cell. The B cell synthesizes immunoglobulins which are inserted into the cell membrane. We can see here the antigen with their epitopes and antibodies with their paratopes. Antigens are from outside and B cells are here. When a B cell differentiates into a plasma cell, the immunoglobulin is secreted from the cell. Antigen selects the complementary antibody on the surface of a B cell. Antigen binds to the surface of the antibody, forming an antigen-antibody complex. The B cell divides and differentiates into plasma cells. The clone of plasma cells secretes the antibody. So the differentiation of B cell into plasma cells. The CH4 domain of the membrane-bound IgM is associated with two other transmembrane proteins known as Ig alpha and Ig beta. The cytoplasmic portion of Ig alpha and Ig beta have two precisely spaced tyrosine residues that constitutes an immunoreceptor tyrosine based activation motif or ITAM. Binding of a specific antigen to the variable region of membrane bound immunoglobulins causes oligomerization of neighboring immunoglobulin molecules. This activates an intercellular tyrosine kinase LYN and some other cytosolic tyrosine kinases. Lin phosphorylates the ITAMs of Ig alpha and Ig beta, and phosphorylated IgMs act on docking sites for SYK. The SKY, another tyrosine kinase, is activated, and activated SYK phosphorylates the tyrosine residues of some other target proteins. Phosphorylation of target protein generates signals that stimulates the B cell to divide and differentiate into plasma cell. The plasma cell further synthesizes the secreted form of immunoglobulin instead of the membrane-bound form. This change occurs due to alternative splicing of heavy chain HRNA. You can see here the structure of Flynn, Lin, and Black. You can see here the items also. Ig alpha and Ig beta sequences also, including the antigen. Primary response. There are millions of beta lymphocytes, which each B lymphocytes, each synthesizing one particular antibody. Some immunoglobulin molecules are present on the surface of B cell lymphocytes. When an antigen enters for the first time, there will be some immunoglobulin molecule on the surface of some B lymphocyte having a complementary antigen binding site. This binding site, the primary response, we continue here. This binding stimulates the B lymphocyte to convert into plasma cell and divide. The plasma cell secretes the immunoglobulin into circulation. And this is known as a primary response. It is weak and short-lived. The secondary response, or the stronger one, is after first encounter with an antigen, some B cells are converted into memory B cells. If the same antigen is encountered again, memory B cells are activated into antibody producing cell. This is called as secondary response, and secondary response is quick, strong, and long lasting. You can see a number of days and weeks and level of antibodies. So in first infection of the primary response, the peak of the antibody is around up 2 weeks for primary response. And when the secondary infection occurs for the same antigen, the level of antibody increases very quickly and very high. Immunization is a process by which active immunity against an infection is produced by administering a vaccine containing a small dose of antigen. This produces a primary response. If the same antigen enters again, it is destroyed quickly by the sensitized lymphocytes. Immunization is used to prevent a variety of infectious diseases. The vaccine containing the antigen may consist of killed microorganisms known as typhoid vaccine, cholera, SARS polio vaccine, live attenuated microorganisms like Sabin's polio vaccine, BCG vaccine, etc. Some toxoids also like tetanus toxoid, diphtheria toxoid, etc. Pure antigen like recombinant hepatitis B vaccine containing hepatitis B surface antigen only. There's one process known as passive immunity. A short-term protection against any infection can be provided by administering pre-formed antibodies. These antibodies may be obtained from animals, human beings, or synthesized in the laboratory, like the monoclonal antibodies produced by hybridoma technology. Monoclonal antibodies are clones of each other. They are used for diagnostic and therapeutic purposes. They are produced in the laboratory by hybridoma technology. A hybridoma cell is a hybrid cell prepared by fusion of B lymphocyte and myeloma cell. B lymphocytes are antibody forming cells. A given B lymphocyte is dedicated to form one specific antibody. Myeloma cells are immortal cancer cells. Fusion of B lymphocytes with a myeloma cell produces immortal hybridoma cells, synthesizing one particular antibody. When the hybridoma cells is put in a culture medium, it goes on dividing forever and does not secrete and goes on secreting the antibody forever. A given B lymphocyte synthesis only one specific antibody. Since only one B lymphocyte has fused with a myeloma cell, the hybridoma cell secretes only one particular antibody known as monoclonal antibody. Monoclonal antibody, a mouse is inoculated with an antigen. The B lymphocytes are taken from its spleen and are fused with myeloma cell in the presence of PEG, polyethylene glycol. Fused cell are selected from unfused cell by culturing them in hypoxanthine, aminopterin, thiamidine medium. You can see here diagrammatically, as I was telling you, injection of pure antigen into the mice, removing the spleen cells from the mice, adding myeloma cells, fusing them with PEG, selected hybridoma cell in het medium, and we have our antibodies. Next is the complement system. It acts in concert with immunoglobulins. It complements the action of immunoglobulins, and it consists of a group of protein present in plasma, etc. Complement system C19 to C1 to C9 and factor B and factor D, etc. The complement proteins are proenzymes which are converted into enzymes by a cascade of reactions similar to blood coagulation cascades. There are two complement pathways, classic complement cascade and alternate complement pathway. The classic component cascade includes recognition phase, recognition unit made up of C1Q, C1R, and C1S participate in this phase. Activation phase, which activation unit made up of C1, C2, and C3 and C4 takes part in the phase. Then the membrane attack phase, which consists of C5, C6, C7, C8, and C9. During the cascade of reactions, one complementary component act on the next, splitting a small peptide off and converting the proenzyme into an enzyme. The small peptide split off is denoted by the suffix A and the larger fragment by B. The enzymatically active fragment is shown by a horizontal bar on it. The next is recognition phase. Binding of at least one IgM or at least two IgG molecules to the surface antigen present on a foreign cell attracts C1, which binds to the constant region of the immunoglobulin, and C1 is activated to C1s bar. Diagrammatically, we can see here antigen with the antibodies coated over them in the cell membrane. The response produces C1QRS with calcium being in the medium. There's an alternate complement pathway. The alternate complement pathway does not involve antigen antibody interaction. It does not require C1, C2, or C4. It requires rest of the complement system and factor B and factor D. In the presence of a bacterial endotoxin, plasma proteases split C3 into C3A and C3B bar. A protein factor B binds to C3 bar to form a complex. The factor D splits B into BA and B bar, and again C3B and CB bar splits into C5 and so on. The membrane attacks complex is formed as in the classic complement cascade, leading to lysis of the target cell. Cell-mediated immunity. The cell-mediated immunity is the function of lymphocyte. T lymphocytes are of four types: cytotoxic T cells, killer cells, helper T cells, suppressor, and memory T cell. T cells have transmembrane receptors that can recognize specific antigen. A T cell receptor is a protein which is made up of an alpha chain and a beta chain joined by a disulfide bond. Each chain has two extracellular domains, a transmembrane region and a short cytoplasmic tail. The alpha 1 and beta 1 domains are similar to the variable region of the Ig light chain in heavy chains, the Bl and H forms. The alpha 2 and beta 2 domains are similar to the constant region of the Lg light chain and heavy chains, known as Cl and Ch1. The antigen binding site or cleft is formed by the alpha and beta 1 domains. The T cell receptors have a diversity similar to Ig diversity. The diversity arises from a gene rearrangement. The gene for the variable region of an alpha chain is constructed from one of the 70V segments and one of the 61G segments. Thousands of different combinations are possible. The gene for the variable region of beta chain is constructed from one of the 52B segments, one of the 2T segments, and one of the 13J segments. Thousands of different combinations are possible. The MCH proteins and MCH genes. The T cell receptors cannot recognize a free antigen. The antigen must be attached to a cell and it should be a self cell. If a T cell is transferred from one person to another, it will not recognize the antigen. The T cell receptor recognizes a foreign antigen combined with a self molecule. These self molecules are proteins known as major histocompatibility complex or MHC complex. The MHC proteins are present on the surface of all the cells and are unique in their individual. MHC genes, MHC proteins are encoded by MHC genes. MHC genes are divided into class 1, 2, and 3. The class 1 genes encode MHC protein 1s, which are present on the surface of all the cell. Foreign antigen combined with self MHC1 proteins are recognized by cytotoxic T cells. MHC class 2 genes encodes MHC2 proteins which are present on macrophages, B cells and follicular dendritic cells. Foreign antigens are combined with MHC2 proteins and are recognized by helper T cells. MHC class 3 genes encode complementary proteins and perhaps some other plasma proteins. MHC proteins. MHC proteins are transmembrane proteins. They have a peptide binding cleft on their extracellular surface. MHC proteins are transmembrane protein. They have a peptide binding cleft on their extracellular surface. They are synthesized on the endoplasmic reticulum. They pick up fragments. They pick up fragments of protein formed by intercellular degradation of exogenous and endogenous protein while on their way to the cell membrane. The fragments are bound firmly and displayed in the surface of the cell. If a T cell finds a foreign peptide bound to an MHC protein, self-mediated immunity comes into operation. MHC class 1 proteins are made up of a large alpha subunit and smaller beta units. The alpha subunit has three extracellular domains and a transmembrane region and a cytoplasmic tail. Alpha 1 and alpha 2 domains form the peptide binding cleft. They display fragments of protein degraded by proteasomes. Cell membrane we can see here which MHC class 1 proteins and antigen fragments. We can see here the peptide binding cleft also of the antigen. MHC class 2 proteins are made up of almost equal sized alpha and beta units. Each subunit has two extracellular domain, a transmembrane region, and a cytoplasmic tail. The alpha 1 and beta 1 domains form the peptide binding cleft. MHC class 2 proteins binds fragment of protein degraded into intercellular vesicles called endosomes. These include proteins of pathogens that reside in endosomes and extracellular pathogens and protein taken up by the cell of endocytosis. These proteins are degraded by cathepsins after fusion of the endosomes with lysosomes. Newly synthesized MSC class 2 proteins binds the peptide fragments and travel to the cell membrane. MHC class 2 proteins get inserted into the cell membrane by displaying the bound peptide on the surface of the cell. Functions of cytotoxic T cells. Cytotoxic T cells destroy the infectious cells along with the pathogen present inside them. This prevents the spread of infection to healthy cells. Most viruses and several bacteria reside and replicate in the cytosol of infected cell. Their proteins are degraded by proteasomes. Peptide fragments are displayed on the surface of the Peptide fragments are displayed on the surface of the cell by MHC class II proteins. The foreign peptide bound to a self-MHC protein is recognized by a particular cytotoxic T cell. The T cell receptors binds to the peptidic MHC1 complex. We can see here diagrammatically. A transmembrane protein CD8 is also present in cytotoxic T cells and act as a co-receptor. CD8 is made up of an alpha and beta chain linked to each other by a disulfide bond. The extracellular portion of the CD8 binds to the A3 domain of the MHC class 1 protein.